Okay, so now let's look at this cytoskeleton. Okay, what's this cytoskeleton? Cytoskeleton actually they are organelles, okay, and their function is just more of them are most to the what we call the support of the cells. Okay, for example, try to imagine in the cells that without cytoskeleton, what will happen is how you change the shape of the membrane. As if, for example, muscle contraction, how you change the shape of the cells. Like for example, amoeba carry out phagocytosis. How they can carry out phagocytosis without changing the shape? So it means that they must involve a certain mechanism. Okay, so we're going to learn these three kind of the three main kind of cytoskeleton filaments. They are proteins. Okay, basically. So who they are? So they are microfilaments, microtubules, and intermediate filaments. And our focus will be in it will be in microfilaments and microtubules. Okay, so based on the structures, let me discuss about these microfilaments. When you look at the microfilaments, the idea of microfilament is they attach to the plasma membranes and involve in the shape of the cells. Okay, they involve changing the shape. Okay, intermediate filaments involve in terms of the locations of the organelles, or fix the location of the organelles, or basically they act as the anchorage of organelle. You try to imagine, guys, okay, we have a nucleus, right? We have nucleus inside the cells. You try to imagine if a jump, so the nucleus will vibrate, correct or not? But to fix the positions, we need the intermediate filament. Is something like when you want to deliver something, I mean, you pack a nice thing already, so you don't leave your things inside a box, right? You put the bubble wraps, right? You put something to stabilize your item before you send it out, right? So intermediate filaments act the same. Microfilaments involved in two important generally two important parts here. Involved in what? Involved in terms of the transport, like vesicle movement, okay, involved in terms of the mitosis. We're going to learn about this later. Okay, so based on the explanation just now, talk about microfilaments, microtubule, intermediate filaments. So can you try to label them? So I assume you guys have done the labeling. So again, guys, how you identify the structure. So first of all, you can see the organelles. So these organelles, they actually attach or stabilize, okay, or anchored by these filaments, right? So these are the intermediate filaments, okay? Intermediate filaments. So they is something like support. Make sure that the organelle actually at the correct positions, okay? So second, you can see that this cytoskeleton attached to the cell membrane, right? So because of this cytoskeletons, they will be able to change the shape of the cell, right? You change the shape of plasma membrane, basically will change the shape of the cell as well. So basically here the answer would be microfilaments, okay? Microfilaments. And the one that form a tube-like structure, okay, allow the organelle to move. It's something a railway track actually. Allow the organelles to move. It's something to move along. So this one actually a tube. So microtubules. Okay, microtubules. Okay. So we continue with some structures and the functions. Okay, so sorry. Okay, so look at this structure. We do have the microfilaments. Something happened, right? Okay, can see. Okay, so we have microfilaments, microtubules, and intermediate filaments. Our focus will be on these microtubules more. Okay, because the next part of the topic we're going to learn about this microtubules. So in terms of structure, no need to remember. In terms of diameter, also no need to remember. Okay, diameter. So these are the diameters of the microfilaments, microtubules, and intermediate filaments. So not in our syllabus to remember, but generally you can see that it forms a tube. Okay, for microtubules. So how to form the microtubules? Basically, they do have this what we call a subunit. So this subunit is called tubulin subunit. So you have the alpha and beta tubulins. Okay, so let us look at it. So what is a protein subunit here? So actin, actin subunit. So this one, we're going to learn it in more detail when come to the muscle contraction in your A2, okay? Muscle contractions, we'll learn them again. And then how about this? We have alpha and beta tubulins, okay, that form this. So guys, you can see that we can actually make it longer. How to make it longer? Add more alpha and beta tubulin. We add tubulin, make it longer. How to make it shorter? Remove. Okay, we remove the tubulin, make it shorter. So this is how microtubules. So we join them together to form a hollow tube. Okay, hollow tube. Okay, how about intermediate filaments? Intermediate filaments actually depends on type of the cells. So that is, for example, in keratins, okay, the intermediate filament keratin can be found in the epithelial cells. Neurofilaments, okay, can be found in the neural cells. Okay, so in our syllabus, you no need to focus in the intermediate filament at all, guys. No need, because our focus in the A2, we're going to look at the microfilaments. In AS, more to the microtubules. Okay, but we need to know their functions. Okay, so what are the functions of microfilament? As I say, that they involve in the shape of the cells. So as long as anything to change the shape of the cells, then microfilaments involved. So for example, in terms of the cells, how the cell can move. For example, amoeba. So how amoeba can move? So cell motility. So it means that we change the shape of the cells. For example, you can see that amoeba. So how amoeba actually move? So they can extend their pseudopodia, and then after that they retract the pseudopodia, then they can move. Can you see that? So this is how the movement involves the change of the shape. So changes of the shape in this A, and then muscle contractions. Also, you need to change, right? Cytoplasmic streaming. So allow the cytoplasm to move, like something you mix. Try to imagine that you have a bag of the drink. You want to mix them, so you press it, right? So you change the shape, and also forming the cleavage furrow during the cytokinesis, because initially your cells like this, you need to change it, the shape. So form a cleavage furrow like this, right? So that later it will form two different cells. Okay, so this is microfilaments. Microtubule quite widely, okay, I mean in terms of their functions. So let us look at what are their functions. So first of all, they involve the cell motility, but different kind. For example, in terms of cilia and flagella. Okay, not the pseudopodia. It's nothing to do with the change of shape, but the beating of the cilia and the beating of the flagella. Okay, so they serve as a spindle microtubule for the chromosome during the mitosis. With this one, we're going to learn it in detail when we come to the mitosis. So how they function, formation of the centriole. So later we're going to learn about this, and movement organelles in the cytoplasm. We're going to learn this one in the next topics. Okay, how the vesicle. Still remember, guys, vesicle, the transport vesicle, move from the RAR to the Golgi apparatus. From the Golgi apparatus, exocytosis take place. Right? Those movement of the vesicle, movement of the organelles require microtubule. So basically, microtubule is something like the railway track. Imagine that it's a railway track. So it means that the organelle will move along this track to the positions that they want to go. Okay, intermediate filaments is quite simple, as we know that they involve in the anchorage of the nucleus and other organelles, so that they have the fixed position. But if they want to move, can or not? Can. Those organelles can move. So how they move? They move along the microtubule. Okay, particularly exocytosis and endocytosis. Okay, so if we stain the cells, you can see that we use a fluorescent dye to stain the organelles. Then you can see that the nuclei labeled as a pink is the nuclei and the nuclei. You see that two cells. Okay, and you can see that the microtubules, the protein tubules, okay, are labeled with a yellow. Can you see that microtubule with a yellow color? And microfilaments, right, are labeled as blue. Can you see that? So you realize that microfilament attached to the cell membrane. So that's why at the cell membrane there you can see a blue color, and then you have the railway track, right? The tracks. So this track actually is the microtubule. Okay, so we have another example here. Can you see that? So the green color here, if I ask you, what is this green color cytoskeleton? So this green color cytoskeleton, you can see that they attach to the membrane, right? So the green color actually is the microfilaments, okay, microfilaments. And you can see that a lot of these anchor the nucleus, right? So this red color actually is the intermediate filaments that anchor the organelle as well as the nucleus. Okay, so with this, I've done for the cytoskeleton.